And today's topics are about analyzing DNA. Okay, so if you've ever wondered, how can they fingerprint somebody with DNA? If there's DNA at a crime scene, how do we know like that that's actually truthful and it actually works out and we can pinpoint a suspect that way? How can we tell that two people are related, right? How can we do things like decipher the actual genetic code? You can't see those A's, T's, C's, and G's, which are just chemicals. How do we figure out that pattern, that code? Because it's been done. We've done the entire human genome. We know all of the bases, all those letters for the entirety of the human genome. Right? And today, I'm going to describe a process where we kind of first did that. But in reality, in modern day times, we have ways and techniques that are even faster than what I'm going to show you today. Okay? But this gives you a good overview of sort of how it's done. So essentially, this is known as DNA fingerprinting in sequencing. The sequencing bit is reading off all the letters. Okay? The fingerprinting bit is trying to figure out, like, did this person commit the crime? Like, does their DNA match this person's? And the techniques are pretty similar, but they're a little bit different. And we're going to simulate one of these down in lab at some point. I don't know if I'll have you guys or the other group, but we'll just see who the heck's here. So let's dive in here. The first step to pretty much all of these things is called PCR. It stands for polymerase chain reaction. Okay? And it uses a big machine, like this one in the picture here, known as a thermocycler. So thermo, heat, cycle, cycle. This thing basically, what its job is it takes a small amount of DNA and it amplifies it. So let's say you've got a crime scene, okay? Or let's say that you want to figure out who's related to who. All you need to do is to get like a swab of somebody's cheek, or maybe a little bit of genetic material, some skin cells or something that was left behind at a crime, a little bit of blood, something that's not, doesn't have to be a lot of material. Okay? But we need a lot of material to do any tests on it. So you've got to make more first. And that step is called PCR, polymerase chain reaction. You're going to see all these words on the test, I guarantee you. If you want to take notes, you're more than welcome to. So essentially, I can break the process down for you really pretty quick here. You've got DNA. How many strands are on a DNA molecule? How many strands? One or two? Oh boy, we're not awake this morning. Two, right? There's two sides to a DNA molecule. Okay? In order to copy DNA, your cells, as well as our machine here, has got to break it apart. It's got to split both sides of the ladder. Now, one way to do that is to heat your DNA up. So this machine thermocycles your DNA. It's going to heat up the little bit of DNA that we have. All the DNA will pop apart, will spread apart, and then it's going to use very specific enzymes to build a new strand onto each of the old template strands. So then we'll have, one copy of all the DNA that happened to be in there. There's probably more than just one cell's worth of DNA in there, right? Even if you have just a little bit, this process can literally make 1 billion copies of your DNA in a matter of hours. Now that's slow in comparison to some of the stuff that we can do more modern times, like there's other techniques and things, but this thing can make like 1 billion copies in like 4 hours or something. It just keeps heating up, rebuilding the DNA, and then cooling down, and it just keeps rotating through this process over and over again, okay? You might run, I don't know, like 30 to 40 times it might cycle through, and when you're done, you've got 1 billion exact copies of your DNA, okay? So this is the first step, okay? The first step to this, PCR, make more copies of your DNA. That will be a test question, okay? Let me introduce these things. These are called restriction enzymes, okay? We use these a lot in all sorts of genetic techniques, genetic engineering, trying to figure out whose DNA is whose. What restriction enzymes do is that they cut your DNA sequence at a very specific pattern. You're going to read about and you're going to see one known as EcoRI, okay? Eco because I believe it comes from E. coli. We get some of these from bacteria. In fact, there's whole catalogs of these that a geneticist can actually order. Like a biologist could order, I need a restriction enzyme that will cut DNA specifically at these bases. Like EcoRI, looks for a sequence GAATC, and it's got to read GAATC, and it will cut it at a very certain location if that code is in the DNA. Okay, so check it out. Your DNA is really long. There's a lot of bases. So the chances that there's a GAATC on there are pretty high. Probably in multiple places along your DNA, these things exist. In fact, it's going to be that for everybody's. It's going to be that for like any animal or any creature that we're talking about. Okay, so what we can do is we can use a restriction enzyme in all this DNA that we just PCR'd and cut it at very specific locations, like on the code. We can get somebody else's DNA and also cut it at very specific locations on the code. Right? Those aren't going to be the same places if they're not the same person, because the DNA, the code is going to be a little bit different from person to person, especially in regions of the DNA that don't code specifically for something. Okay, seeing how we're going to kind of play this out? You make some cuts at known places, and if different people have different spots like that, you'll end up with some different lengths of DNA. So now the next step is we've got to figure out how to see this DNA, because even though we've made billions of copies of it, like, this stuff is virtually invisible. It's so tiny, right? So how do we actually map it out? That step is done through something called gel electrophoresis. Another word you're going to see, we're going to kind of simulate this downstairs in like sort of a pen and paper lab. So what we do, and you might have seen these before, is we use something known as a gel, okay? And we're going to sort our DNA fragments by length. So remember, the restriction enzymes, it cut it at the same place every time, but that spot, that section of code, was in different locations down the string. And it's going to be different for different people. Okay, it's going to be different for different organisms. So what we do then is we take that DNA that we've cut, and we put it in one of these wells at the end of this. Gel has little holes cut in it. It almost feels like, it kind of feels like Jell-O. Okay, we have this stuff downstairs. In fact, if you take AP Biology, you will do that. You will physically do this. I've got some pictures in here from an AP Biology class doing exactly this. So what you do is you put the DNA that's been cut in these wells. Okay, maybe first well contains DNA from the suspect. Okay, suspect 1. Okay, next well contains DNA from suspect 2, and the last well contains the DNA from the crime scene. Then all we have to do is apply electricity to it. Check this out, the DNA is negatively charged. What happens when you've got a negative charge and a positive charge? They're going to attract, right? Just like two opposite sides of a magnet, opposites attract. So we electrify this gel with a negative over here by where we put the fragments, and a positive down here on the other end with electricity, and the fragments start to migrate through the gel. So you put all this DNA over here, and it starts to trek its way down the gel. Now the gel's kind of thick and hard to get through. So the smaller the piece, and this is a test question right here, the smaller the piece, the farther it goes. Aha, are you seeing it now? So if your DNA has been cut at exactly the same spots as the DNA we found at the crime scene, we've got a match, because the little fragments are going to go exactly the same length. Now we still have to dye it, okay? So usually we have to dye it and expose it like to UV light to get the bands to show up that you see in it, because DNA is quite clear stuff. Okay, so it'll migrate down a certain distance. We expose the gel, and then we can see it. So a lot of bits and pieces have to be right here. You got to use the right enzyme to cut it. It's got to be an enzyme that will find locations in there. You had to run PCR to make more of it. Okay, you've got to run the gel for a certain length of time, because if you keep leaving that electricity on there, all the fragments are going to end up at the bottom. You want them nice and neatly spread out so you can analyze them. And so there's a technique to it. Okay, it's almost an art. And Mr. Chastain will tell you, like in his AP Biology class, some students will just nail it and do a really good job, and others will end up with a total mess. Like their gel won't turn out at all. Right? So there's a lot of little techniques to this thing. Here's one from our class. This is down in our lab. Here is a gel setup. So this little plastic thing here is the gel, where the gel would go. Okay? There's a buffer solution in there. It kind of floats in this liquidy stuff. And then you can see the positive and the negative terminals on this thing. So we're going to push the DNA fragments across the gel. And these are two gels that came out. And you can see there's little indents at the top of the gels that have been cut out. That's where the students pipetted the DNA into. Okay, and then they ran the gel and it migrated downwards in here. So tell me again, which fragments went the farthest, the small ones or the big ones? The small ones. Awesome. It's definitely a test question. So now we just look across it and we try to line them up. Okay? It's not going to be a match unless it's a perfect match. Right? If it's a perfect, I mean, you could have similar length pieces of DNA that you cut between two different organisms even. But if it's a true match, every single piece will match. So these bands, if you will, represent not just one piece of DNA, they represent billions of pieces of the exact same length piece of DNA. That's the only way we could see it if we had lots and lots of it there. So that's kind of how gel electrophoresis works. That's how PCR works. Those two steps are going to be somewhat the same for a lot of these things that we're going to talk about. So let's look at that again here. If I ran this gel, because you might see a test question something like this, which fragment on this gel, A, B, C, or D, was the smallest? A, B, C, or D? Did somebody say D? Yes, D. D would be the smallest. It went the furthest. You've got an arrow there even showing you, okay? DNA has a negative charge because of those phosphate groups. And so if we put the negative here and the positive here, all the DNA is going to migrate down the gel. And we're almost always going to set it up for you like this. Okay? So that's going to be important here in just a second. So remember that kind of fact. So here's a short little video clip. This is time lapse. See it separating out? The little ones travel the farthest. Okay? It started at the top and it migrated downwards. Okay. Happened there. Come on. Uh-oh. Okay. All right, I think we're back on here. So you've got in this situation, you've got here's a simple DNA fragment, like from a fingerprint, from a crime scene. So we've got sample 1, which is the crime scene, sample 2, which was the suspect, and sample 3, which is maybe the shop owner. Okay? So we want to know if this person did it or if the DNA we picked up happened to be the shop owner's DNA. Can you see it? What DNA did we get from the crime scene? Was it the suspect's DNA or was it the shop owner's? It's the shop owner's, isn't it? Like every single band had better match if it's a true match. Okay, so looking at the suspect, some of the bands do match, right? Not surprising. We're both humans. We're all humans here. So there's going to be some similar sections of DNA, but there's going to be some that are different. In the different sections, the crime scene that we're studying better have all of those if we've got a true match. Okay, so I put a little step-by-step here for you. First step, we PCR it. We make more. Now, the second step, we use restriction enzymes to cut it at known spots. Okay, we cut the suspects, we cut the shop owners, we cut the crime scene sample. Okay, then the fact that the DNA fragments are going to be different lengths allows us to run a gel electrophoresis and spread them out. Then we just look for matches. It's simple as could be, right? Just as an aside, I was called to jury duty one time. It was a really terrible experience. I never ever want to do it again. It was very sad and depressing for me because one of the questions they were asking the potential jurors, they were asking us, do you believe that DNA fingerprinting is an actual way that you can identify a suspect? They were asking us this. And I, of course, said, well, yeah, like, duh. And that immediately precluded me from being on the jury. Like, they didn't want me because I accepted scientific reasoning. They wanted somebody on the jury who, didn't accept that DNA actually worked, that we could actually do this, right? It's like, you explain it like this, it's almost dirt simple, isn't it? But they were trying to say, well, the suspect couldn't possibly be there, even though we have his DNA at the scene, because DNA technology is really doesn't really work that well. Like, you can't accept it. It doesn't work in court. Well, that's absolutely untrue, right? We all know that. But they were trying to get the jury set up in such a way that it would be better for their client. So anyway, jury duty. So here's another crime scene. Here's the DNA from the crime scene. Who did it? A, B, C, D, or E? C. Okay, yep, I think that's right. Yep, C is the one who did it. See, even though there's some others that have matches, it's the one that matches perfectly. Like, when you say we've got somebody's DNA at the crime scene and you do this, like, it's not like just a little bit matched. Like, you've got a perfect match. So yeah, it really does work. So here's an AP biology class down in our lab working. You can see them there with the pipette. This girl here has got a pipette. She's dropping the DNA into these wells right here. Okay, it's dyed, and then it's going to migrate once they apply electricity to it down the gel. Okay, it's a very difficult process. You got to be really careful. There's some serious technique involved. So let's talk about how we sequence DNA. That's how you fingerprint somebody. Okay? In fingerprinting, you don't care about all the exact code. Notice how we didn't do ATCG blah 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 blah. Okay? We did, here's a chunk, it's that long. Here's a chunk, it's that long. Do they match the chunks from the other person? In this scenario, we're going to do some of the same stuff. The only difference is we want to know precisely what does the code read. So as you can imagine, this process is a little bit more involved. Like, you wouldn't just do it quick for a crime scene. You're trying to physically figure out the entire sequence of DNA code. Okay, so first step's pretty similar. You're going to run PCR. You got a little bit of DNA. You got a swab from somebody. You got a little sample from some fish tissue or something. And you run PCR to make more DNA. Okay? Once you've got the DNA and you made all this stuff, you've got to make four test tubes up. Okay? Check it out. How many bases are there in DNA? A, T, C, and G, right? I'm holding 3 fingers. There's 4 bases in DNA. Okay? So you make 4 test tubes. And you're going to put some of this DNA in each of these 4 test tubes. So we're going to have a test tube labeled A, one labeled T, one labeled C, and one labeled G. Because we need to figure out where on the code those letters actually fall. Does the code start with an A? Does it start with a T, a C, or a G? Is the second base a G, a C, a T, or an A? Like, where does it fall? So the next step that we need to do is we're going to put nucleotides in here. So remember, a nucleotide is just a single base with part of the ladder on it. Okay, that's how DNA is built. Okay, so anyway, when you split apart the ladder to build the new ladders onto the old ladder pieces, you need nucleotides. Now, normally we supply that in the PCR, and we're going to do that again, only this time we're going to put some specially tweaked nucleotides in there. Not a lot of them, just a little bit. Okay? We're going to put some specially tweaked A nucleotides in there. And these A nucleotides, when they bond onto the DNA strand, okay, what's going to happen is when they get incorporated in, the enzymes that build the new strand will terminate. They will stop after that weird base gets put down. They won't go any further. So what we're going to end up doing is making fragments of DNA that end at every A. Fragments of DNA that end at every T, that end at every C, and end at every G in 4 separate test tubes. Okay? See where we're going here? Once we put this stuff that's been essentially cut at every single base into the different wells, we run it with gel electrophoresis, and the very first letter is going to end up where? At the very top of the gel or at the bottom? It's 1 base long. Think about how long that is. It's 1A. Okay. It's going to end up at the bottom of the gel. So let's kind of take a look here. We've put these special nucleotides in here. They're labeled DD, like up there. and they terminate the DNA strand. So if you're trying to copy this stuff, and let's say you're using, I don't know, G here, it would copy C, A, and when it put down that G, dunsies. Can't go any further. So you got a short little segment. How long is it? It's exactly the length up to the very first G. So I put all those in the same well, so we're going to have to label our wells in this one. In the crime scene, it didn't matter. Now we're going to have a T, a C, an A, and a G well, and we're going to migrate it. So knowing that, the smallest pieces go the farthest. So the piece that's only one base long, it went almost all the way to the bottom, didn't it? The piece that has two bases, it went next closest to the bottom. Now what's the base on the end of that second piece? So let's just check this out. If we're talking about fragments here and reading this thing, I'm going to read it from the bottom up. I can literally read the DNA code. Okay? So well T went the farthest. So I would start with a T. The first base of my DNA code is a T. What's the next piece? What's the next base? You see it? It's a little hard to see. A is the next one down. Okay? So this fragment down here, this A fragment, if I could actually physically see it, what it would have on it would be TA. It has the T and it has the A because it cut off at the A. Okay? The next piece up is this other T. Well, that little piece right there, if I could actually read it, it's longer than either of those two pieces beneath it. It actually contains a TAT on it. See it? Like you can't read that, but we know it does because that's where it landed. The next piece up is a C. It's in the C well. So it was cut off at that very first C. So this piece here contains TATC on it. It's a little bit longer than the pieces that are beneath it. And so we just read this thing, and you can just physically read it off. TATCCACTG. There's the code. That's slick, right? This process used to take a really long time. And now we have, like I said, nowadays we have techniques that go a lot faster. So let's read this off. This is just like a test question. If I gave you this DNA sequence and I said, I want to know the actual DNA code. What's the code from this sample of DNA? What would it be? How would you read it? Can you see? You'd start with bottom or top? Bottom. This one would be C, C. What's this one? G, T, 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 A, T, G, C, A. Ta-da! It's like so simple, right? What a tricky way to kind of do that, though. Like think about the engineering and the thought that had to go into that. Like we can't read the DNA; it's too small. How do we got to make more of it first? So we make a whole bunch of it. Well, how do we know like where the letters fall? Well, what if we cut it into segments that terminate at every single letter, and we run them down? The smallest pieces will go the farthest. It's really slick, right? So remember, we talked about two different things here. We talked about DNA sequencing, which we just showed you here, the actual code, right? And we talked about DNA fingerprinting, like just hacking the DNA up at known locations and seeing if it matches somebody else's. Okay, they're really similar processes, but like in this one, you got to have 4 wells, an A, a T, a C, and a G. Actually, not to confuse you, but usually there's a 5th well. Usually they put another one over here with some known stuff in it that will migrate specific lengths, and it will help them read it. Like they can read it, there'll be a line over here, and it will say like 50,000 base pairs. And they know like if you've got one there, you got a length of DNA that's exactly that long. And so now we can compare like against even different creatures and things. So it's kind of, it's more technical than what I showed you here, but that's the basic idea. Okay?